Hey guys, my name is Alex Carroll, and my presentation is on the reversible transdifferentiation of blood vascular endothelial cells to a lymphatic-like phenotype in vitro. This was discovered by Dr. Lynn Cooley and other scientists at the University of East Anglia in London. They then reported it in the Journal of Cell Science. These particular scientists were actually involved in the study of anti-cancer drugs. Their goal was to halt the capillary formation in tumors, therefore making it impossible for them to grow and spread throughout the body. However, their research ended up leading them to discover that the cells involved, those of blood and lymphatic vascular systems, are able to switch their genetic profile by turning off genes expressed by the blood endothelial cells, which I'll call BECs, and turning on those expressed by lymphatic endothelial cells, which I'll call LECs. Actually, here's a picture that you can see of a capillary that they grew in the lab. You can see through gene staining that this blood vascular cell is showing genes that are coded in pink and orange, which are supposed to be in LECs. So that's just something you can look at. This whole discovery actually resulted from the analysis they were doing on human umbilical vein endothelial cells, adult human aortic endothelial cells, and adult human dermal microvascular endothelial cells being grown within several in vitro models. These models consisted of a variety of monolayer cultures and 3D matrices. Those within the 3D matrix environment formed tube-like structures instead of the normal flat structures. So they performed a microarray analysis in order to compare gene expression and found an altered expression of a number of genes previously identified to be expressed only in BECs or LECs, but never in both. Those genes caused the BECs to transdifferentiate to lymphatic-like phenotype. They then proceeded to further studies to see if this genetic change was dependent on a 3D development and found that it also occurred in 2D mitrogel assay and on top of a dense collagen gel in monolayer fashion. However, they did not do so in a monolayer growth on top of fibrin gels. Instead, they kept their flat cobblestone appearance. Tests of dependence on tubulogenesis were then done that concluded that even when this process was inhibited by the presence of a broad-spectrum matrix metalloproteinase inhibitor, the transition to a lymphatic phenotype still occurred. They then decided to take cells that had transitioned and return them to a monolayer culture on fibrin and found that they reverted back to their original BEC expression. All of these experiments led to their conclusion that gene expression is directly regulated by the extracellular environment of the cells and not by specific signals during embryonic development. Also, they believe that the traditional well-established types of monoculture assays they used might in fact represent processes involved with the formation of lymphatic vessels rather than that of blood vessels. So they hope that this important breakthrough will lead to the discovery of new revolutionary treatments of cancer from all around the world. I too hope that this arises and that our medical battle with cancer will finally make that major advance that we need to finally cure it. So that's my cell bio news. Hope you guys have a good day.